organizer for this great opportunity to present. I'm going to talk about actually on my Li Wang, student at the University of Southern California, and another piece of work was done at a research laboratory Shaz group. I just heard that there was a very similar work in methods by Kaplow. So if you compare those two work, you're welcome to find their paper in Nature Method. So RNAi is a very throughput technology. It allows us to investigate the silencing effect of although this technology is really great, it does come with multiple issues. For example, due to the tolerance to mismatch between the RNAi probe and the mRNA, usually we observe unwanted silencing effect of other genes, and this is called off-target effects. And also due to the limitation of the RNAi delivery system, we often see innate immune response. And often those immune response does interfere with the biological process under investigation. And also not every gene can be effectively knocked down by RNAi. For example, if the gene's expression is very low, it's very difficult to knock down effectively. Also, you order RNAi probes from different providers, you often see very inconsistent results. So definitely there's some difference between the RNAi probe design, and that is still a problem. So to really overcome those issues, current methods are either try to define better probes or to design better statistics to make calls on the screening readouts. Actually, I realized that there is a special session this afternoon to focus on RNAi data analysis. So if you're interested in those topics, definitely you should want to stop by. And another commonly taken approach is that to run just another secondary screening based on the primary screening to achieve better accuracy. And if you figure out certain genes that are interesting and are validated by those high throughput RNAi, then you better run some low throughput validation experiments. Follow up. All these over time and relatively different way. I try to for this system influence needed to download and then develop this additional information. Does Brimmed is trying to integrate PPI and also the pathway information as such a knowledge base. So the two in MC genomics, genome research, until they integrate those information or information, make a assumption. Assumption is that real hits are not randomly protein network. Instead, in the local subnetworks or pathway guilt by rule. So for this hypothesis, those red nodes represent hits, and our assumption, we would like to think those in this subnetwork are more likely to be true compared to isolated. Is that things involved in the same biological process, they would have more interaction with it among them. So the first study, we look at 24 genome-wide RNAi screening, and those were obtained from the FLY RNAi database. The Drosophila protein-protein interaction network was obtained from the string that contains over 10,000 proteins and 200,000 interactions. So the first step we like to do is to really test our whether so for each RNA screen, look at how many hits for that, how many interactions we see, and then we want to just estimate how significant those in. We basically run two patient tests, randomize the level of those RNA, calculate actions are, and in the same patient test, we just have RNA hits. We are permutate those interactions. They preserve the degree those two p-values are basically in the sense that one is very significant. Screening experiments significantly connected into RNAi hits. And I just show one, actually, they're color, but the projector does show it. We think one reason why this particularly highly connected hits for biological process really hard to readouts. Although we don't have hands-on, didn't do those experiments by ourselves. Biological process is very tough to read out, and we think this is one of them. And basically, none of the reasons I believe that could be the case. As we need to ranking algorithm so that the more reliable will be ranked on top, and vice versa. Given a node that we want to rank, we basically to flag the relationship between the query nodes in the network that with known headers. In total, we consider 12 algorithms or ranking functions of them, but basically we consider 4 types of pairwise, and then we also consider 3 methods, which we summarize the pairwise similarity scores, and by combining them, in total we have 12 different functions. Because we don't know for each gene whether it should be a real hit or not, so there are no straightforward way to test which whether our ranking algorithm indeed works or which function works better. So we have to devise an alternative approach. Instead, we just design a rank-based algorithm. So for example, for specific screening results, we obtain a hit sets denoted by those red nodes, and we have a non-hit set. So we just select one node from this hit set and put it into this non-hit set, and just pretend it as a negative hit. And we call it a simulated false negative. Then we run whatever ranking algorithm on this set of genes, because we know that this is a positive hit. So presumably a good ranking algorithm should rank this node on the top. Of course, sometimes we know that some hits in these sets could be actually false positive. So a good ranking algorithm actually should rank it to the lower or the middle part list. But if we do this ranking for every gene in this set, then on average, we expect a better performed ranking algorithm should put this set of genes near the top. So this is the results that we obtained by looking at 4 genome-wide screening results. You can see that the diffusion kernel-based method combined with the third serialization method gives us the best performance in ranking false negative nodes. The direct neighbor method does poorly. But I mean, although I said poorly, but you can see that it's still quite good in the sense that it ranks on average there's false negative near the top of the list. For each specific, this figures screening experiments, what's the best is. So process, usually the best are different. However, the top performed viral replication, and for this lowest biological process, nuclear import still gives the best performance. But that makes us to think that the ranking is not due to the algorithm, but low quality of the RNAi hits. Seems to suggest that this particular experiment, the readout is really of low quality. Extreme case, suppose that all the RNAi hits, basically then no matter what the kind of rankings, you won't be able to get a good ranking. Validation example of this particular experiments, we have some reproducible data available, and we can see that there is a strong positive correlation between our ranking score and this reproducibility rate. So that if this score is high, usually you see a much higher reproducible rate. This is also true for another screening experiment Wnt signaling pathway, although the trend is not that strong as this one, but it's still significant. We did multiple other validation, and I just show one example. And I move on to our second part, which used pathway information instead of screening. Genes using insulin display in murine and genes either is or decrease the knock down by design a pathway extension, find the raw screen results, and to insulin signaling components. So you overlap this RNAi hits on the pathway for this particular pathway. The multiple hits can be a KEGG pathway, and it's intuitive to that the paths that link the hits could be the end of the line that signaling was transduction. However, if there exists multiple that link the hits, then we really don't pathway which path was used by the signal. So you have to basically consider all the paths that link those RNAi hits. But if we have another hit interact with those, then we'll increase our confidence to tell us that a certain subpath and know those paths are really involved in section. Because this is a partial screening, so we see on those paths and we run a whole genome screening. You'll see that clear signaling pathway knows on that path be the positive hits because there's negative in the RNAi. So this is how the algorithm works. We obtain this list of that's list in the screening experiment. Then we all the KEGG pathway. Basically, we try to identify those ways that you see multiple hits, and then we identify possible paths that link those RNAi hits. Those paths, these steps seeding, and then we try to expand context of global protein-protein interaction network and see other RNAi interact with those seeding. If we do see some interaction between RNAis with this signaling with this signal, just attach a nose to this module, and we step expansion. And the final step, we just try to prune those sub that contains no RNAi hits, so we can obtain a PET module. Basically, using that 100 hits and each step out looks like. And this is the final output, and the module. So those rows are the original, and those nodes are original experiments, but we and we also include nodes colored by the green, but they were basically based on the it was a hit, as they could be potentially positive hit. Really justify algorithm one tell the experiment designed so that for sure when for the original screening in research rank at the 9th position experiments. Insulin signaling pathway that tells us that RNA information. And after algorithm, we see that although it's won by one position, that's because there are were over. So in this raw hit result, it's about 30. Query the pathway, and you should increase the signaling pathway. Successfully pull hits can be cast together subnetworks or pathways based on multiplied RNAi screening data, and we to integrate such information, or at least we demonstrated that they are helpful for providing the true hits. We think that of other types could be helpful, and that would be future works. The first of the work was funded by NIH and NSF. The work was supported by the Merck Research. Finally, I would thank the audience for your attention.
Yes, our insulin screen was focused on pretty much to expect there to be more interactions among the protein. If you have a more unbiased screen, how big would the RNA be for you to get vision of RNAi hits clear, so that you could use your hit action? Kinase phosphatases or cancer or trackable G need to be?
Actually, we would prefer screening be unbiased, but at that time when the experiment, the RNAi technology was still at its early stage, so we don't have the resource to do whole genome wide. But back to your question, I think even for genome-wide screening, I think the KEGG pathway is definitely useful information, and definitely you can consider other information as well. I think that the algorithm can handle pretty large number of things, although we showed on several hundred genes, but if you run the...
Well, I think it would probably work. I mean, it would seem that it would work better the bigger the screen is, because you would perhaps get more clustering. But if you have a less wide screen, for instance, the druggable genome or cancer library, which is maybe a couple thousand or so siRNAs, would that still be big?
Your question is if this algorithm can handle several thousand hits, is that the question?
No, I mean, if you have a small screen, then it's very less likely that the hits would cluster together on the protein-protein interaction network. So you wouldn't derive benefit from mapping onto the network, perhaps. But if you have a bigger screen, then it's more likely that they would cluster together, that you would get all the...
Right.
Yeah.
Yeah, I agree. The first approach, the ranking algorithms, they don't ask you to basically cluster those nodes together. That's not right.
No, I mean clustering, meaning in the sense that are in the same area of the protein interaction network.
Yeah, maybe it is more offline.
So since you're looking at enrichment protein-protein interaction networks and pathways that have previously been ignited, obviously this is going to more highly prioritize hits that are involved in areas that have already been studied. Do you have any advice for screeners who are searching biology as to how they would counteract that bias?
I've been working in the pharmaceutical area. Actually, people would like to ask me to provide more reliable, basically hits by multiple even literature evidence. So that's not a problem for my purpose. But for your purpose, if you're look at the novel pathways or novel interaction, novel hits, that's not been previously heavily studied. So there's not many interactions available. I think one solution is that you just add your own. You just generate your own, even hypothetical interaction could be and then try to information into the network and see if that will help you to expand your. That's one kind of way to test it. So basically, the efficiency then I don't think can really help you to get really novel things from empty.
This is a question. When you're testing your ranking algorithm, I see you had hits a set of non-hits, and then you tried putting some of the hits into the non-hits rank. I was wondering how you originally got those hits and non-hits, if you figured out exactly what those hits and non-hits are.
I mean, those hits are based just on the original screening results. We do not say this is a or this is a non-hit. It's purely experiment results. And for those hits, there came some false positive. So some of them could be just, they're not supposed to be hit.
If there are no further questions, the speaker and all the speakers in the session. Thank you.